All right, welcome back. So last time we left off talking about the axial skeleton. Now we're going to change gears a little bit and we're actually going to switch back to the nervous system. So remember that previously we talked about the central nervous system, which includes the brain and the spinal cord. And now we're going to talk about the peripheral nervous system. So the peripheral nervous system is everything outside of the spinal cord. So it includes things like spinal nerves that we'll talk about in more detail later in the week. And it also includes things like your cranial nerve that we'll talk about next time. So one of the things that the peripheral nervous system does is it helps carry out the orders, if you will, of the central nervous system. So it actually relays information, let's say motor information, for example, from the spinal cord out to the muscles and causes the muscles to contract. And similarly, it relays information from our extremities, so from our arms and legs, from our trunk, from our internal organs, relays that information back to the spinal cord, which then goes up the cord back to the brain so that we can make meaning from that information. So the peripheral nervous system then includes a variety of sensory receptors. So they give us information again about ischemia, so lack of blood flow to our internal organs, stretch of our internal organs, but then also more I guess less exciting sensory information about whether something is touching me or not, temperature, deep pressure, that kind of information also travels over the peripheral nervous system. So then move on if it'll let me. There we go. All right, so in terms of sensory receptors, so they obviously respond to a variety of different stimuli. So they respond to changes in the environment. And an important thing about their ability to respond is that there has to be an adequate stimulus. So the temperature change has to be sufficient to cause the opening of ion channels to allow for depolarization of those neurons. And so once they depolarize, then they send an action potential, if we're talking about an afferent, up that axon, back to the spinal cord, and again, back to the brain eventually. So there's three different ways to classify sensory receptors, and you can see the first one there is classification by stimulus type. So what is it that causes them to depolarize? So you've got different types of receptors. So the first one on there is a mechanoreceptor. Most of the ones that we're going to spend our time talking about are mechanoreceptors. So as you would guess, they respond to mechanical stimulation. So really deformation, a change in shape of those receptors causes them to depolarize. So for example, the receptor there pictured in the lower right is one called a Pacinian corpuscle. And so those are found in joint capsules, for example. And so when the joint moves, so when you flex your shoulder or something like that, it's going to cause compression on the anterior part of the capsule and a stretching on the posterior part. And so that'll cause a deformation, particularly on the posterior aspect, because the anterior aspect of the capsule is going to have some slack in it. But the posterior aspect of the capsule is then going to be tight. There's going to be some tension in it. And so then that tension will cause a change in shape of the Pacinian corpuscle's receptor here. So that's the receptor region, which will then cause the opening of ion channels and then an action potential to be generated that goes up the cord back to the brain so that your brain knows where your shoulder is in space. Other types of mechanoreceptors we're going to talk about include Golgi tendon organs, or just tendon organs, that are responsive to the amount of tension in a muscle, or specifically in the tendon of the muscle. We'll talk about muscle spindles that are responsive to muscle stretch or not, and then a variety of other receptors. Thermoreceptors, as you would guess, again, are responsive to temperature changes. So particularly extremes of temperature, which can then fall over to the nociceptors at the bottom that we'll talk about in a minute. So thermoreceptors sense changes in temperature, hot or cold. Photoreceptors, photo means light, and so photoreceptors are in the retina of the eye, and so they respond to light, so they're part of the visual system. Then we've got chemoreceptors, which are responsive to chemicals. So some of the ones we'll actually talk about in the next class, in Structure and Function 2, we'll talk about chemoreceptors that you have in your aortic arch and in your carotid arteries, which are the two big arteries on either side of your trachea. And so those chemoreceptors monitor the blood. And so they monitor specifically things like the pH of the blood. So is the blood too acidic? They also monitor the partial pressure, or just for short, the pressure of carbon dioxide in the blood, the pressure of oxygen in the blood, and then we can respond to fluctuations there. So if the pressure of oxygen goes down, we'll increase our breathing rate, we'll increase our heart rate. If the pressure of carbon dioxide goes up, we'll do the same two things. If your pH of the blood goes down, so it gets more acidic, same type of thing. And so those are examples of chemoreceptors. And then nociceptors. Nociceptors are pain receptors. So noci means harm, and so they are receptors that are that generate action potentials in response to extremes of hot or cold or excessive pressure. So like a really deep pressure, if you get a crush injury or somebody, really grabs you hard and compresses your skin and the muscles underlying it, then that will cause nociceptors to depolarize. So the first way to classify receptors then is by what causes them to send an action potential. Another way to classify them is based on the location of that stimulus. So it's classification by location, but all of these receptors are inside of the body, or at least in the skin, right? So they're inside of you. So when we talk about location, we're actually talking about the location of the stimulus. So is the stimulus outside of the body? Those are exteroceptors that sense stimuli outside of the body. So examples there include receptors in the skin. So you have receptors inside of your hair follicles that are receptive, well, redundant, but that are receptive to deflection of the hairs. And so they let you know that something is touching the hair and thereby probably touching your skin. So again, the stimulus there is external to you. Something outside of you is touching you. Sorry, my dog is scratching and jingling his collar, as you can probably hear. There we go. All right. And so anyway, exteroceptors pick up things outside of the body. Interoceptors are responsive to stimuli inside of the body. So the example I just gave you of the chemoreceptors in the aortic arch and in the carotid arteries, those are interoceptors. So they're monitoring the blood, but also special sensory organs, again, muscle spindles, that monitor the length of muscles or the rate of change of that length. So the muscle shortening, they're receptive to that as well. So that would fall under interoception. It's something inside of the body, a change in muscle length that's causing them to depolarize. And actually, speaking of muscle spindles, so they actually cross over a little bit, and they are also proprioceptors. So proprioception is generally thought of as position sense. So it's where you are in space. And so your proprioceptors are all essentially a special type of interoceptors, but they include receptors in the muscles, so your muscle spindles, receptors in the tendons, so tendon organs, and then also receptors like Pacinian corpuscles in the joints and ligaments that let us know where we are in space. So which part of the capsule is stretched or not, which muscle is stretched or not, and then which ligaments are stretched or not. And we use all of that information then to determine where we are in space. So is my shoulder flexed? Is it in a neutral position? Is it externally rotated, etc.? We know all that based on the information coming back to the brain from a variety of proprioceptors in the muscles, in the joint capsules, in the ligaments, etc. And then a third way to classify receptors is based on their structure. So one of the things to differentiate here at the outset, talking about the structure, is our general versus our special senses. So should probably talk about what the special senses are, because those are the ones we're not really going to talk about. So the special senses include vision, hearing, equilibrium, which means balance. So that includes the receptors inside of your inner ear that let you know whether your head is moving in which direction. And then your smell receptors, so olfaction, and then your taste receptors as well. So those are all your special senses. So again, vision, hearing, equilibrium, smell, and taste. And then everything else is part of the general sensory system. So on the general side, most of the receptors are essentially modified dendritic endings. So remember, thinking back to chapter 11, when we talked about the basics or the fundamentals of the nervous system, we talked about the basic structure of a neuron. And so the neuron, remember, has dendrites, which receive information from somewhere, and then they relay that information back through the body of the neuron, down the axon, and then eventually out to the axon terminals. So these general sensory receptors then, their receptor that initiates the action potential is effectively just a special type of, or just a modified dendritic ending. So it's a modified dendrite, essentially. So in terms of those general receptors then, so there's basically two ways to subcategorize them. They're either unencapsulated or encapsulated. So the unencapsulated ones, what that refers to is, does that modified dendritic ending have a connective tissue capsule around it? If it does, then that's encapsulated. And if it does not, then that's an unencapsulated receptor. So the first one there is a free nerve ending. And so what makes it free is the fact that there's no connective tissue capsule around that ending. So the free nerve ending, because there's no capsule, it's just a dendrite hanging out there, right? And so free nerve endings are present almost everywhere in the body, but there are lots and lots of them in your skin and also in your connective tissue. We didn't talk a ton about connective tissue, but connective tissue is also all over the body. So we talked about connective tissue in terms of the wrappings of a muscle. And it's going to come up again here when we talk about the wrappings of a nerve in a little bit. But connective tissue is found around your organs, in your abdominal cavity, in your thoracic cavity. It's found underneath your skin, etc. So free nerve endings, lots of them in your skin and lots of them in your connective tissue. And what they do is they respond primarily to painful stimuli. So changes in temperature, particularly heat above 120 degrees Fahrenheit or cold below 40 degrees Fahrenheit, are going to cause them to depolarize. And when they do, then that signal, when received by the brain, is interpreted as pain. So extremes of temperature cause free nerve endings to depolarize. Also, low pH, so acidity, causes them to depolarize. So that could be, in your skin if you get acid on yourself, but it could be also an acidic environment inside of the body as well. So, for example, if you exercise at a really high intensity, so if you do, let's go with, you run a 400 or an 800, you're going to generate a lot of acid as a byproduct of producing energy for that. And so then that produces a pain sensation, right? And so that change in pH then produces that pain sensation as the tissue gets more acidic. That causes pain, or that causes free nerve endings to depolarize, which then the brain perceives as pain. They're also responsive to certain chemicals. And they are particularly, and they being the free nerve endings, they are particularly sensitized by inflammatory chemicals. So, for example, if you get a crush injury, so something smashes your skin, or if you get a cut, something like that, you have a special type of cell, particularly a lot of them in the connective tissue, around blood vessels that are called mast cells. And so mast cells, we talked about them some. We talked about the little bit of connective tissue that we talked about. But what they do is they release this soup of inflammatory chemicals, and one of them is histamine. And so what histamine does is it sensitizes the free nerve ending, so it makes it easier for them to depolarize, makes it easier for them to send electrical signals. So it hypopolarizes them. Again, it's going to move their, trying to tie this back into previous talks, it's going to move their resting membrane potential a little bit more positive. So that's what histamine does. So your free nerve endings are extra sensitive, meaning that let's say you got a cut and it has scabbed over, but you'll notice that the area around that cut is red for a few days after the initial injury, and it's particularly sensitive. So if you start poking at that skin, that's going to hurt a lot, right? And so because of that soup of inflammatory chemicals, particularly histamine, that's going to sensitize those free nerve endings. So just any little bit of pressure causes them to depolarize and causes that pain signal. So those are your free nerve endings. So what I want you to know about free nerve endings, the big thing I want you to know is that they send mostly pain signals. The Merkel cells and discs, you can see one of the things in anatomy is a lot of structures were named for the scientist, the physician, et cetera, who first discovered them, first named them, those kinds of things. And so Merkel cells here are an example of that. But you're also seeing alternative names that don't include, scientists or doctors, et cetera. And so the alternative name there is the epithelial tactile complex. For me, sometimes the scientist names are actually easier. So at any rate, Merkel cells are found in your skin as well. And so those function as light pressure receptors. So if something is just barely touching your skin, some kind of soft touch, then that's going to cause the Merkel cells and Merkel discs to generate an action potential, which then the brain perceives as light touch. And then we have our hair follicle receptors. And so those are special free nerve endings that wrap around the hair follicles. And so they're responsive to light touch. So if something just barely deflects your hairs, then that is going to cause those special free nerve endings, the hair follicle receptors, to depolarize so that something's touching you. So that's how, where those come up is like if a bug is crawling on you or something, or a mosquito lands on you, usually they deflect your hair follicles a little bit, or deflect your hairs a little bit, which causes the hair follicle receptors to depolarize, and so then you know that bug is on you, and then you can flick it off of you. And so here's more information about each of those things. So you can see the free nerve endings at the top, the Merkel discs there in the middle, and then the hair follicle receptors at the bottom. So as you can see, this is table 13.1 from the textbook. But in case you didn't buy the textbook, I put it in the slideshow. Or in case you didn't rent the textbook, I put it in the slideshow. So there's that figure. All right, so those that we just covered were our unencapsulated receptors. So now the ones that are left are the encapsulated nerve endings. So again, these are still basically modified dendritic endings, but now all of these dendritic endings have some sort of connective tissue capsule around them. So for example, I'll click over to the next slide. So this is also table 13.1, but you can see that the capsules change shape a little bit. So this one, I forget how this one is described as being sort of like a egg-shaped. This one's described as being egg-shaped, the Meissner's corpuscle. And then this one kind of looks like a cut onion because there are so many different layers. This one, the Pacinian corpuscles have as many as 60 layers of collagen around them. And then we'll talk about the other ones. All right, so first things first, the tactile or Meissner's corpuscles. So again, these are found in the skin, but you find lots of them in areas where you've got hairless skin. So the follicle receptors are responsive in part, along with the Merkel cells, are responsive for light touch in areas of skin with hair on it. But places like the bottom of your feet or your fingertips or the palms of your hand, those don't have hair on them. And so we need to be receptive to light touch there. And so how we do that, or the receptor responsible for doing that, is largely the Meissner's corpuscles. So the Meissner's corpuscles do light touch in areas of hairless skin. So again, bottom of your feet, palm of your hand, those kinds of places. And then the Pacinian corpuscles, so where we find those are also in the skin, but in addition to being in the skin, we find Meissner's corpuscle, Pacinian corpuscles in the periosteum of the bones, also in tendons, in ligaments, and in joint capsules. So one of the things to know about Pacinian corpuscles is that they are responsive to deep pressure or vibration or stretch. So deep pressure really applies in the skin. Vibration or stretch applies in the case of joint capsules or ligaments. But the important thing about the Pacinian corpuscles is that they're only responsive to that stimulus when it's first applied. So they are rapid adapting. So they whenever there's a change, so if something is, if you, let's go back to the earlier example, if you flex your shoulders, you pull your arms straight up, the Pacinian corpuscles, again, on that posterior part of the shoulder capsule are going to depolarize right as that happens. And then once you move to your new position, they're going to stop sending signals. So really, they make the brain aware of changes in position or a new deep pressure that wasn't there before. That's as opposed to the Ruffini endings or the bulbous corpuscles. So they do, they're in the same basic places. So you find those in joint capsules as well, but they're also in the skin. So they respond to continuous pressure. So we'll talk about phasic versus tonic receptors. And so the difference between those two is that your phasic receptors respond, they're rapid adapting is probably the best way of putting it. So they respond to the change as it happens, and then the tonic receptors are either slow adapting or non-adapting, meaning that they may send signals all the time. And so why that matters is that we use these two, particularly in the joint capsule, so the Pacinian corpuscles and the Ruffini endings, to determine, they're really important proprioceptors is what I'm trying to say. So again, when I change position, the Pacinian corpuscles are going to generate action potentials, letting the brain know that we've changed position, and they code that information. So if I move my arm slowly, they'll only depolarize a few times per second. But if I move my arm really fast, then they're going to send more action potentials per second, so the frequency increases. Remember, that's how the nervous system codes for the intensity of a stimulus. The more intense the stimulus, in this case, the faster I'm moving, the more frequent depolarizations you're going to get. So the Pacinian corpuscles then send information, I'm moving right now, and then how fast. And then once I've stopped moving, then the Ruffini endings, the tonic receptors, are going to send constant signals. So let's say you leave your shoulder there. So let's say you have raised your arm, you flex your shoulder and you're just going to leave it there for 30 seconds or a minute or something. And so once you've done that, once you've stopped moving, then the Pacinian corpuscles stop depolarizing, but now the Ruffini endings depolarize again on that posterior part of the capsule, and they're going to send constant signals, 1 or 2 action potentials per second back to the brain to let it know that, yep, posterior part of your shoulder capsule is still stretched, so the brain then interprets that as meaning that my shoulder is in a flexed position. And again, we're going to get a variety of other information. So the brain will also be getting information from muscle spindles in the biceps and the triceps to let it know that the triceps is lengthened, the biceps is shortened. And so combined with the information it's getting from the Ruffini endings, that is how the brain is able to interpret where your limb is in space. So the thing to know about the Ruffini endings then is that they are tonic receptors. So they send constant signals. They are responsive to continuous position or continuous pressure. So another example of those, and I don't know that we need one, but why not? So again, both the Pacinian corpuscles and the Ruffini endings are found in the skin. So whenever you go to sit down, you're going to get an initial response whenever you first hit the chair from the skin overlying your glutes and overlying your glute max and overlying your hamstring musculature. That skin is going, the Pacinian corpuscles in that skin are going to initially send signals letting the brain know that there is pressure. And then once you have been seated for a few seconds, then the Pacinian corpuscles shut off, and then the Ruffini endings let the brain know that there's still pressure there. So you're still seated, there's still compression of that skin overlying your glute max and your hamstring musculature. So the brain interprets that as you're sitting down. All right, so the muscle spindles. We'll talk about them again in a couple lectures, but the most important thing I want you to know about them is they are responsive to muscle length and then the rate of change in muscle length, if that is in fact changing. And so there's actually 2 subtypes of muscle spindles or 2 different types of fibers in the muscle spindles, and we'll talk about that more in detail later. But muscle spindles include both phasic and tonic receptors. So they let the brain know that the muscle is changing length and how fast, and then once the muscle has stopped changing length, they let the brain know how stretched it is or not based on the frequency of depolarization. Tendon organs, I'll probably refer to them repeatedly as Golgi tendon organs, just know I'm talking about the same thing. So tendon organs, as you would expect, are in the tendon, and they are sensitive to tension, so being pulled apart. Because effectively what happens is when the muscle contracts, then that's going to put tension on the tendon of the muscle. And so the Golgi tendon organs have fibers that actually wrap around the collagen fibers, where they're dendrites, if you will, actually wrap around the collagen fibers, and then they're encapsulated. And so when there's tension in those collagen bundles, that compresses those tendon organs and then causes them to send signals back up to the spinal cord and brain, letting the brain know that there's tension in that muscle. So one of the things to know about Golgi tendon organs, though, and I'll talk about this again later, is that when they're stimulated, they actually cause an inhibitory response. So they cause the muscle to shut off. So they act as a sort of safety valve to keep the muscle from generating too much tension, and they cause it to relax. All right, so then same thing, here's 13.1. All the same stuff I just talked about. And this is in the slideshow in Canvas as well, if you want to take some time to read that, or obviously you could just pause the video. But there's all the receptors I just talked about. All right, so let's talk about sensation and perception briefly. So the difference between the two is that sensation is awareness of a change in your environment, and then perception is interpreting that awareness, right? So the example the book gives, and I'll steal it, is that if there is a rock in your shoe, then, let's say it's under your big toe, then you are going to sense that, hopefully, as a deep pressure under your big toe, and then, so that's the sensation. That's recognizing that there's something there, that there's an external stimulus, so that compression of the skin. And then the perception of that is probably discomfort, right? And so because of that perception of that as being uncomfortable, I'll probably do something about it. I'll take off my shoe and, drop the rock out, those kinds of things. So there's a term on there, which we talked about a little bit, but I need to clarify, especially as we get into the autonomic nervous system later. So the somatosensory system, so remember, anything somatosensory deals with the body. So that's going to deal with the body wall and the limbs. So basically everything that's not, let's leave it at that. Let's go with the body wall and the limbs. So in terms of the levels of organization, so the first one on there is the receptor level, and we've talked about a lot of this stuff actually, in terms of generating a signal. So that's initiating that action potential. So generating the signal is dependent upon the type of stimulus that causes that particular receptor to depolarize. So for something like a Pacinian corpuscle in the skin, deep pressure is going to cause it to depolarize. And so again, that mechanical deformation causes the opening of ion channels, which then allows sodium to come rushing inside, tying this back to chapters 11 and 12. And then that initiates an action potential that goes up that, it's actually technically the dendrite. So it goes up the dendrite to the cell body, through the cell body, and then along the axon. So speaking of that, then we move from that initial neuron, that first order afferent, and I'll explain what that means in a second. From that first order afferent, we're going to move up the spinal cord, and then into the brain, and then from the thalamus in the brain up to the somatosensory cortex. So there's some parts of the circuit that I should probably explain at this point to make this make a little bit more sense. So remember that afferents, as opposed to efferents, afferents relay sensory information. And so you have what are referred to as first, second, and third order afferents. So the first order afferent includes the receptor, so something like a Pacinian corpuscle or a muscle spindle, and then all of that neuron that it's attached to, because remember that the receptor is basically just a dendrite. So that depolarization goes up that dendrite back to the cell body. Now, one of the things that is important to be aware of is that these are really long cells. So initially, when I was taking a very similar class to this as an undergraduate, I figured that cells are microscopic, and so there was just kind of this continuous relay of electrical signal from one cell in a nerve in my big toe, all the way up through, I don't know, I figured probably thousands of cells to get back to the spinal cord. But that's how it works. You've got one cell with a really long, so sticking with the big toe example, one cell with a really long dendrite that goes all the way back to the spinal cord. So again, that cell could be 3 feet long or longer, depending on how tall you are. So it's a really lengthy cell. All right, so dendrites go all the way back to the spinal cord. The cell body for that sensory neuron actually sits outside of the spinal cord in something called the dorsal root ganglia, which we'll talk about later. So relays the signal back to the cell body that sits right next to the spinal cord. That signal moves through the cell body out to the axon of that neuron, and then to a second neuron, probably in the spinal cord at that level. The second neuron that relays the signal up the cord to the brain is called the second order afferent. And then the third order afferent takes the signal from the second order to the somatosensory cortex. So you can kind of see that going on here. It's not the best graphic for describing first, second, and third order afferents, but for example, this muscle spindle, so it's going to send its sensory information. It'll actually synapse in the spinal cord with a second order afferent, and then the second order afferent, so the second neuron, if you will, then relays that signal over to the cerebellum. So muscle spindles can have only a first and second order afferent, or sometimes they'll have a first order afferent that goes up here to the brainstem. So this is that's a really long cell. So that cell might be 4 or 5 feet long. So it goes all the way up to the brainstem, and then it synapses with a neuron in the brainstem that sends the signal to the thalamus. So this is the second order afferent here, and then the thalamus synapses with another neuron in the motor cortex, and that's our third order afferent. So in terms of first, second, third order afferents, the short version is first order afferents bring the information back to the cord, sometimes to the brainstem. Second order afferents often bring the information to the thalamus, and then third order afferents bring the information from the thalamus to the somatosensory cortex. And so here I'm referencing, remember back in chapter 12, we talked about the primary motor cortex, which is up here, and then the primary somatosensory cortex, which is back here. And so that's throwing it back to chapter 12. That's where we recognize where something is touching us, or where that sensation is coming from. So if a muscle is being stretched, or skin is being stretched, or whatever, we recognize where in the body that's happening in that somatosensory cortex. Remember, that's where we had those really funny looking pictures of the body with really big feet and really big hands and faces, but relatively small, like arms and legs, to depict how sensitive certain parts of the body are. So that's really the circuit level. So relaying the information up the cord. And then once we get to that somatosensory cortex, that's the perceptual level. That's where we make meaning of that information. So something is touching me on the backside of my hand, for example. So yeah, we'll stick with that. That's good. All right, so that's that stuff. So let's move on to hopefully make this a little bit more interesting. All right, so first order afferents. So remember, first, second, third. So the first order afferents bring the information from the body back to the spinal cord, and occasionally all the way up the cord to the brainstem. So there's 4 groups of first order afferents. And so the way they're classified is, as you can see there, based on the diameter of the axon, so how big it is, and how myelinated it is. And so the diameter of the axon and the degree of myelination both determine the speed of conduction. The bigger the diameter of the axon, so the wider its cross-sectional area, and the more myelinated it is, the faster the electrical signal that it conducts. So in terms of the 4 groups then, the way they're set up here is, so you got your A alpha fibers, and those are the largest and the most myelinated. So they conduct their electrical signals really fast. The group C fibers have the smallest cross-sectional area, or the smallest diameter, and they are unmyelinated, so they conduct their signal really slowly. So from top to bottom, it goes from really fast to really slow. So your A alpha fibers, so what are we talking about here? Again, those first order afferents are going to be connected to, or the receptors are actually part of them. So in this case, we're talking about things like muscle spindles. Yeah, let's go with that. So information about how much a muscle is stretched or not travels over a type of neuron, an A alpha neuron. And so what that means from a practical standpoint is that neuron has a really big diameter and is well myelinated. And so from a practical standpoint then, Information about muscle stretch or change in length travels back to the cord and up to the brain really, really fast. So how fast? About 300 miles an hour. Those neurons are capable of conducting their electrical signal. So really, really quickly. So the type of information that travels over A alpha fibers, again, is information from the skeletal muscles. So are they changing length or are they not? And then how fast? And then, so that's afferent information. Also, efferent motor output travels over A alpha fibers. So whenever we cause muscles to voluntarily contract, that also travels over A alpha fibers. No, there we're talking about an efferent as opposed to an afferent. But why that matters then is, so think about something like, if somebody's playing football and you got a running back who is running full speed and then needs to change direction really quickly to outmaneuver a safety. And so they got to be running full speed, stop, plant, change direction to go around the safety. And so the brain has to be really aware of where the body is in space, so how much we want to flex the knee when we stop and change direction. And then we have to, in order to actually change direction, cause the external rotators of the hip to contract really quickly. And so we want that sensory information back to the brain as fast as possible, and we want the motor output out to those muscles as fast as possible, so we can change direction, stop on a dime, etc. And being able to do that is dependent upon how fast the signals in and the signals out can travel. So they travel over those A alpha fibers that again can conduct their electrical signals at up to 300 miles an hour. A beta fibers, a little bit different. So A beta fibers are smaller, and they're more lightly myelinated. So their myelination isn't quite as complete as the A alphas. Why that matters is then because they're smaller and because they're not as well myelinated, they can't conduct their electrical signal quite as quickly as the A alphas. So whereas the A alphas can conduct their signal at 300 miles an hour, the A betas, now we're down to 30 miles an hour. So the type of sensory information that travels over the A betas is information about touch. So like some of the stuff we talked about with like deflection of the hair follicles or just light touch in the skin. That type of information travels over the A beta fibers because we need to be aware that something's touching us, but we don't need to be aware of that quite as rapidly as we do the position sense. So A beta fibers then kind of general sensory information from the skin is probably a good way to think about those. The A delta fibers, now we're even a little bit smaller and a little bit less myelinated. They are still myelinated, but not super well. So they conduct their signal at about 15 miles an hour, so about half the speed of the A betas. So the A deltas then are responsive to what's often referred to as fast pain. So if you burn yourself or if you cut yourself or something like that, you're going to get a really quick withdrawal reflex. You're going to pull your hand away if you burn yourself. That fast pain travels over those A delta fibers so that we can get away from whatever it is that's hurting us. Again, we want to be aware of it quickly, but we don't need to be aware of it quite as quickly as we are about where we are in space. So the A delta fibers then, what I want you to know, fast pain. The group C fibers, they are the smallest, as mentioned. They are also unmyelinated, so their signals do not travel fast at all. So putting a miles per hour on it, their signals travel at about 2 miles per hour, so much, much slower. So the group C fibers are responsive or typically relay information about what's referred to as slow pain, so like chronic aching pain. So if you have something like bursitis, and so you're just sitting there in class and your hip is just kind of aching, right? So that information is traveling over your group C fibers. So it's that, like I said, that slow kind of aching pain. So local anesthetics, so like if you go to the dentist and they inject something into your mouth and then you can't feel your face for a little bit, just as an aside, what those do is those block the voltage gated sodium channels. And remember, throwing it back to those earlier chapters, the way that nerve depolarization works is there must be some, usually it's a chemically gated channel, depends upon what type of thing we're talking about, or let's go with that. All right, so there has to be a chemically or mechanically gated channel that opens up, causing sodium to rush inside, and then once you hit that threshold voltage, that causes the opening of those voltage-gated channels, which then allows that wave of depolarization, also known as an action potential, to move along the dendrite back to the cell body or down the axon. But what local anesthetics do, so that numbing agent that the dentist injects into your mouth, what it does is it blocks those voltage-gated sodium channels, so you can't initiate that wave of depolarization. So all of the pressure of the dentist, poking around and doing whatever it is that they're doing in your mouth, that nerve is trying to initiate that signal. So the mechanically gated channels on some of those sensory receptors in your mouth are opening, but that nerve can't open its voltage gated channels, so it can't relay the signal back to the brain, so the brain then can't perceive it as pain. So that's what the local anesthetics do. Cryotherapy is cold therapy, it's ice. So, whether it's an ice bag or you stick your hand in a slush bath or something like that, that's all cryotherapy. And as you're probably aware, if you put ice on it, it reduces pain, whatever it is. And so the reason for that, it's actually not entirely understood, but there's two pretty likely theories. The gist of it is, if you put an ice bag on your elbow or something for 20 minutes, after you're done, it actually takes effect after about 7, typically goes numb, you're not usually able to really feel that part of your arm anymore, right? And so what happens there is that nerve conduction, so the ability to send electrical signals, slows by about 1/3. And so that's probably because that cooling doesn't allow the ions to move as fast as they otherwise would at a normal temperature. And so you just can't send as many signals back to the brain. And so if the brain isn't getting as many pain signals traveling over the A delta or C fibers, it doesn't hurt as much. And so the way that ice works then is it doesn't allow the pain nerves to depolarize to send quite as many electrical signals. And so the brain doesn't perceive as much pain anymore. And then something else that ice probably does is causes vasoconstriction, so it causes the blood vessels to close down, to decrease their diameter. And in doing that, of course is going to limit blood flow to the neurons, to the axons of the neurons, so they can't get back to their resting membrane potential quite as fast, because in order to send a bunch of electrical signals to generate a bunch of action potentials, they're going to have to pump sodium back out and pump potassium back in, and that takes energy. And so if you reduce the amount of oxygen that's able to get to the cell can't produce as much energy, so it can't run those sodium potassium pumps quite as fast, and so it can't send as many electrical signals. So ice or cold therapy, cryotherapy, probably works through 2 ways, probably by reducing blood flow a little bit, but then also by cooling that tissue, that fluid down, reduces the rapidity of the movement of the ions. And so either way, your pain nerves can't send electrical signals quite as fast, nor can your other sensory organs, right? So like if you start poking on that skin that's numb, you won't really feel that as well either. So your Pacinian corpuscles are less responsive as well. All right, so speaking of pain, this is from Rocky 3, and every time I talk about pain, it reminds me of this, so I'll play this little clip for you. It's quick. Yeah, so like I said, that's from Rocky 3. So Mr. T there, as Clever Lane, has a prediction for Rocky during their boxing match, and his prediction for Rocky is pain. So, speaking of pain, so we talked about a little bit of this previously. So, obviously pain is unpleasant, but pain does serve a useful purpose, as you know. So it warns us about actual or impending tissue damage, and motivates us to try to protect ourselves, to try to get away from that pain. So in general, pain is caused by extremes of pressure. So something's smashing tissue. So like if you get a bruise, that's an extreme of pressure. That's compression. That's crushing of the tissue, or extremes of temperature or chemicals. And regardless of the stimulus, that then is going to cause free nerve endings to generate action potentials, which again, the brain is going to interpret as pain. And again, tying this back into some of the things that we talked about in chapter 12, So one of the things I mentioned when we talked about the brainstem was a structure in the brainstem called the para-aqueductal gray matter, or PAG, para-aqueductal gray. It doesn't matter that you know the name, but we talked about that structure, and I mentioned that it was involved in what's referred to as descending pain control, and that's basically where the brain blocks pain signals from coming up the spinal cord. So it essentially decides to ignore pain signals by keeping them from reaching itself. And so the structure that initiates that blocking of pain signals is that para-aqueductal gray in the brainstem. And so where you see this is, for example, if you know, and I've mentioned this example before, but if you hurt yourself, let's say you tweak your hamstring in a playoff game your senior year, and so you just kind of fight through it to get through the game, and then afterwards, maybe an hour or two afterwards, you realize that the pain is actually much, much worse than it had been before, and much worse than you would have anticipated. And so what has happened there is during the game, you can kind of block that pain out, and then afterwards, you stop doing that. And so this ties also back into the whole first, second, third order afferents. So remember, your first order afferents are going to bring that pain signal up to the spinal cord, and then they're going to relay it to a second order afferent that's going to generally bring the information up the spinal cord. So what descending pain control does is neurons in that PAG run down the cord, and they synapse at that spinal cord level between the first and second order neurons, and they release a signal, or not signal, they release a inhibitory transmitter that keeps the first order afferent from releasing its neurotransmitter, whether that's glutamate or substance P. And so basically, the pain signal can't go up the cord is effectively what happens. And so then the brain never receives that pain signal, and so then it doesn't hurt. And then after the game, you stop blocking it, and so now things are painful for you again. So that's descending pain control. Again, you don't need to know that, just I think it's interesting. All right, so some terms on there: pain threshold and pain tolerance. So pain threshold describes the intensity of the stimulus required to cause the free nerve endings to depolarize. And that's pretty much the same in all of us. So painful stimulus could be electrical shock. It could be extremes of cold. It could be changes in the internal chemical environment. I'll talk about some of those here in a minute. But all of us perceive pain at about the same level of stimulus. We all recognize something as painful at about the same level of stimulus. But pain tolerance describes the highest intensity of pain that a person is willing to bear. So how much pain are you willing to deal with and keep going? And so pain tolerance is markedly higher in athletes than it is in non-athletes. Now, it depends upon what type of athlete we're talking about, but pain tolerance is obviously your contact sport athletes tend to have a little bit higher pain tolerance, but also your interval sprint sport athletes. So soccer players, basketball players, et cetera, where you have to sprint repeatedly, and also sprint cyclists and swimmers because of the discomfort of the training, right? So if you do lots of sprints and then take rest breaks and another sprint, it's going to cause a lot of burning and a lot of discomfort, and you have to do that regularly to be a fairly high level athlete. And so athletes develop a higher pain tolerance. They develop a higher amount of pain that they're willing to deal with. And there's been some interesting studies that have looked at pain tolerance in athletes versus non-athletes. One of the more interesting ones, I think, they kind of went to the extremes in terms of their groups for a study group and a control group. So this study comes from a group of ultra runners from 2013, and the 11 ultra runners that were picked to participate in this study had all run the 2012 Trans-Europe foot race, which covers 2,700 miles, actually almost 2,800 miles, in 64 days with no rest days. So basically running for 2 months straight every single day. And so the people who'd done that, 11 people who'd done that, were compared to 11 non-athlete controls. And so what the individuals were asked to do was to stick their hand in a bucket of ice water. And as you have probably experienced at some point in your life, that's pretty unpleasant. If you have to put your hand or foot in an ice bath, that's pretty unpleasant for a little while. For about, like I said, 5 to 7 minutes, then it goes numb, and then it's kind of whatever. But those first few minutes are pretty uncomfortable. And so both the athletes and the non-athletes were asked to place their hand in this ice slush bath for 3 minutes. So you kind of reach that peak of discomfort, and then they're allowed to pull their hand out. And so from the endurance runners, all 11 of them completed all 3 minutes. So they stuck their hand in there, they left it in there for 3 minutes, and then at the end of that 3 minutes, they were asked to rate their pain on a scale of 0 to 10, 10 being the worst pain they've ever felt in their life. And so on average, the ultra runners said the pain of having their hand in that ice bucket for 3 minutes was 6 out of 10. So it's pretty uncomfortable, but not unbearable. Just something you can deal with, but it's noticeably uncomfortable, right? In the non-athlete group, they were asked to do the same thing, stick your hand in the slush bath for 3 minutes, and then after the 3 minutes is over, rate your pain. And so from the non-athlete group, again, there's 11 of them, 8 of them couldn't even make it to the 3 minute mark. They quit after an average of 96 seconds, and all 11 of them rated their pain after having their hand in the slush bath as 10 out of 10. Worst pain they've ever felt in their life. Which I think part of that too is, if you're a serious athlete, you've probably done your share of cold treatments. And so, part of that too is just acclimatization. You're just used to doing it. You know what it feels like. And some of the non-athletes probably have never done something like that. So they rated it as being incredibly painful. Another interesting study on pain tolerance in athletes versus non-athletes. Actually, this one looked at elite athletes versus pretty good athletes versus non-athletes. It's an old study. It's from 1981. But they took Olympic level swimmers and then club level swimmers and then non-athletes. And for each of the groups, what they did was they put a blood pressure cuff on their arm and then they squeezed it really tight. And then they asked the participant to open and close their hand, kind of like you do if you've ever donated blood, similar kind of thing. And then they counted the number of hand squeezes they were able to accomplish. And so as you can imagine, the more squeezes you get in, the more acidic that environment gets inside your forearm, and so it gets increasingly uncomfortable. And so the elite level athletes, the Olympic level swimmers, were able to get an average of 132 hand contractions, 132 for the Olympic level swimmers, 89 contractions for the club level swimmers, so quite a bit more, and then the non-athletes only managed 70. So the Olympic athletes got almost double the number of hand contractions, so almost double the amount of pain effectively that they were willing to tolerate than the non-athletes were able to. And there's a 2017 study that kind of replicated that, but they used cyclists instead of swimmers. And one of the interesting things that they found was that the pain tolerance indicated by that tourniquet test, so basically the more contractions you could generate, was actually predictive of race performance. And so the gist of it was the more pain you were willing to bear during that little test, the more likely it was that you were going to be a high-level cyclist in that case. And so this comes from a book that's called Endure. It's by Alex Hutchinson, came out in 2018. It's a really good book if you're interested in the extremes of human performance. And so Hutchinson said, quote, "Pain tolerance is both a trainable trait and a limiting factor in endurance." And he goes on to say, "It does appear that top athletes really do push themselves to a darker place and stay there longer than most of us are willing to tolerate." End quote. So pain tolerance, again, it's higher in athletes than in non-athletes, but it's actually the ability to tolerate pain, to no one's great surprise, is actually an important part of being an elite level athlete, being able to deal with the discomfort of the training. And on the cryotherapy thing, I mentioned that on the previous slide, so I won't revisit that. All right. Whoops, let's go back. There we go. All right, so this is my quasi case study. So we're going to pretend that when you got out of your car today, assuming you went anywhere, because but if you'd gone somewhere, you got out of your car and you stood up too fast, you accidentally hit your head on the door frame, so you got pain on your head from slamming it against the door frame, what would you do to immediately decrease that pain? Because your head's going to be throbbing. How do you make that pain go away as fast as possible? And so oftentimes people will say, well, you take Tylenol or you, do any number of other things. But the answer that I'm fishing for here is you're probably going to rub it, right? So whenever you if you slam your fingers in something, you probably shake your hand out. If you, hit your head on something, you probably rub your head. If you're playing baseball and you get beamed, you're not supposed to rub it, but rubbing it would make it feel better. The question is why, though? Because it does, right? So if you slam your head on something and then you rub it, that's going to reduce the pain. But it shouldn't. Like, why would that have any effect on the pain? And so the answer deals with another pain control theory. And again, you don't need to know this, but I think it's interesting, which is called the gate control theory of pain. And the gist of it is that, and this also ties back into that whole first, second, third order afferents, that those first order afferents, both the A betas, which again are going to relay sensory information about touch and hair deflection in the skin, the A betas and the A deltas are going to synapse with the same second order neuron. And so what you've got here, so this, I know, I'm sorry, the graphic is not great, but it's one of the better ones I could find of gate control theory. So this is your A delta fiber up here, this is your C fiber here, and this is your A beta. So again, this one's going to be your fast pain fiber, and this one is going to relay information that something is touching you, right? And so after you slam your head in the door, on the door frame, this neuron is going to be depolarizing a whole bunch of times. So it's going to be sending a lot of signals back to the cord, relay with this second order afferent, and then goes up to the brain. We perceive that as pain. So it's going to be sending lots of signals because you got lots of pain. But then if you start rubbing the area where you have pain, so you start rubbing that spot where you slammed your head, now you're also getting a something is touching me signal. And so the thought behind this gate control theory of pain is basically that whenever you start rubbing the area that hurts, you get kind of an alternating set of signals that come up the cord. So basically you're going to get an ow signal followed by something is touching me, ow, something is touching me, ow, something is touching me. That's as opposed to if you, before you rubbed your head, all you were getting was ow, 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 ow. And so that mix of signals then causes the pain to decrease. So the brain has to process other information from that same area of the body. So it gets, again, tactile information, something is touching me, in addition to the pain information. And so the effect is that we perceive that as being less painful. So it hurts less because you have that additional stimulus. And why does any of that matter? Sometimes athletes in particular are big fans of these things. So there's all kinds of different brand names. So there's Icy Hot, Tiger Balm, Theragesic, any number of others, right? Bengay is another one. But they all do basically the same thing. They're collectively referred to as counter irritants. Most of them have menthol in them. So they're all some kind of a skin cream. And so like Icy Hot, and I think Bengay too, have patches now. So if you go to the grocery store, Walgreens or whatever, they got patches that are usually for low back pain. You stick on there and it'll make your back pain reduce. And so effectively what it does is just what I explained, that let's say you got low back pain, you stick one of those Icy Hot patches on there, the menthol in those patches is going to cause sensory receptors to perceive that tingling sensation. And so the brain then gets this combination of ow, tingle. And so that, again, that combination, rather than just strictly pain signals, causes us to perceive it as being less painful. And so you can see there that Theragesic is recommended for muscle soreness, for backaches, for arthritis. And so again, so if you got, let's say, arthritis in your fingers and you were to rub Theragesic or Icy Hot on there, you're going to get some of that pain, but in addition to the pain, you're also going to get a tingling sensation. So it's not that Icy Hot or Theragesic or any of the rest of them actually affect the inflammatory process. You know, they don't reduce inflammation, they don't reduce swelling, they don't do any of that stuff. Basically, they just cause the brain to have to also process a tingling signal in addition, caused by the menthol, in addition to the pain signal, which the result is that you perceive less pain, so you're able to tolerate a little bit more than you would be otherwise. So that's what those do. All right, case study for you. So we're going to pretend that you're a high school athletic trainer working a baseball game. And in the third inning, your catcher gets run over by an incoming base runner in a play at home plate, depicted there by the Braves guy. The runner lowers his shoulder, hitting your catcher in the abdomen. When you arrive at the plate, your catcher is lying face up, so supine. The catcher is conscious and holding his stomach. He doesn't appear to have any serious head or neck injuries, so you support him, like you help him stand up, and walk with him back to the dugout. Once there, the catcher tells you he has pain in his upper left abdomen. You decide to have him sit out and continue to observe him. As the game progresses, the catcher complains of increasing pain in his left shoulder, as well as feeling dizzy, lightheaded, and nauseous. You also notice that he appears pale, and when you take his blood pressure, that is lower than you would expect with a high pulse rate for somebody who has been resting. So the question I have for you here is, what could be going on? What could be going on with our catcher that's causing him to, after this impact where he gets a shoulder in the abdomen, to feel like he is dizzy, lightheaded, and nauseous, and then to also have pain in his left shoulder? And the answer is that he has a ruptured spleen. So your spleen is in the upper left quadrant, so you can see up here. So your spleen filters out old red blood cells. Is kind of its job. It's the thing that gets injured in. So if you ever had mono and they kept you from playing whatever your sport was while you had mono, it's because mono can cause an enlargement of the spleen, and since it's a relatively solid organ, it's easy for it to get damaged. So you have to, till the mono goes away, until the spleen returns to normal size, you're not allowed to play contact sports. So he's ruptured his spleen, but the real question I have here is, so if the spleen is in the upper left quadrant and it's in his abdomen, why does he have pain in his left shoulder? And the answer to that is that he has, so that's called Kerr's sign, K-E-H-R''S, Kerr's sign. And so that's referred to as visceral, sorry, not visceral pain, it is visceral pain, it's pain from an organ, but it's called referred pain, which is pain away from the site of the actual injury. So he's got, the actual injury is in his left upper quadrant of his abdomen, but his pain presents in his left shoulder. So that's referred pain. So it's pain away from the site of injury. So our visceral organs, so our abdominal and thoracic organs, so your spleen, your stomach, etc., they have receptors in them that are sensitive to stretching of the tissue, crushing of the tissue, but also ischemia, so lack of blood flow, and then potentially irritating chemicals. So we get that like in the stomach. So what's happened here is that he's had a crush injury to his spleen, and so the pain receptors, if you will, from his spleen are then sending signals back to the brain to let the brain know that damage has occurred. But the reason that it presents as pain in his left shoulder and not pain in his abdomen is because the visceral, so the organ, pain afferent signals travel in the same way, or travel in the same pathways into the spinal cord and up the spinal cord as our somatic, so the rest of the body, pain receptors. And so the brain has probably never had to perceive pain coming from the spleen before. And so because that pathway that houses the pain receptors from the spleen is shared with the receptors from the left shoulder, while the brain is getting a pain signal from the spleen, it interprets it, because it's never seen that before, it interprets that as pain in the left shoulder. Because you've probably had left shoulder pain at some point in your life, and so the brain is used to getting lots of signals from the left shoulder. It's never gotten a pain signal before from the spleen, and so it incorrectly interprets that as pain in the left shoulder rather than pain in the abdomen. A more classic one is when somebody suffers from a heart attack. One of the things that they often complain of is pain in the left arm, and you can see these are our referred pain patterns, but you can see for the heart, so if somebody's having a heart attack, they may have pain in the chest, but they also tend to have pain running down the left arm and occasionally up in the neck. So again, because those nerve roots, and we'll talk more about those in 2 times, because those nerve roots and tracks of axons coming back to the brain carry not only visceral pain signals from the heart, but also the somatosensory signals, so sensation of the skin or muscle spindles or of the joint capsules, etc. Those are all traveling the same pathways back to the cord and up the cord. Again, the brain is going to misinterpret that as something in the left arm because it's used to getting sensory information back from the left arm. It's not used to getting sensory information back to the heart. So the brain incorrectly interprets ischemia, lack of blood flow in the heart, as pain in the left arm because it hasn't seen a pain signal from the heart before. And so that's why you end up with those referred pains from damage to the abdominal organs or the thoracic organs. All right, home stretch now. So 2 slides left. This one's about the structure of nerves. So The nerve that most people are familiar with is your ulnar nerve, aka your funny bone. So if you've never found your ulnar nerve before, we've talked about some in class, but if you go back to your medial epicondyle in your humerus, so remember that's that big knob on the inside of your elbow, if you go just behind that, you'll feel a little cord-like structure back there. That's your ulnar nerve. And so ulnar nerves, or the ulnar nerve, but nerves in general, are basically bundles of axons, right? So they're relaying information for the most part back to the brain or away from the brain. So they're just these, like I said, big bundles of axons that are relaying information for the most part to and from the brain. And so I'm actually going to skip down to this and then we'll come back to this. So in terms of their classification, so nerves can either be mixed nerves, which almost all of them are. We'll talk about some that are just afferent or efferent next time. But mixed nerves carry a mix of afferent, so sensory information, and efferent, motor information. So your ulnar nerve is a mixed nerve, so it carries sensory information back to the brain, but it also carries motor information away from the spinal cord, so out from the brain, out the spinal cord, and then out to muscles. So for example, if you were to slam your elbow on something and you were to injure your funny bone, what ends up happening there is probably the most notable thing that you would notice would be that you typically have tingling into your fourth and fifth fingers, so your ring finger and your pinky, because the ulnar nerve is responsible for relaying sensory information from that medial aspect of your hand back up to your brain. And so when you crush it, you cause depolarization accidentally of some of those neurons that are responsible for sensation of the medial aspect of the hand. And so the brain interprets that as tingling or numbness in that aspect of your hand. But if you really smashed your ulnar nerve, you would have also noted that there is motor weakness. So the ulnar nerve innervates the flexor carpi ulnaris muscle, which hopefully you'll remember is a wrist flexor and ulnar deviator, as well as the flexor digitorum profundus muscle, which is a deep finger flexor. And so if you get damage to the ulnar nerve, then you're going to have weakness making a fist because of the damage to flexor digitorum profundus, and also weakness in wrist flexion because of the lack of signal. So not damage to the muscle, but the muscles aren't able to receive the excitatory signal from the brain. And so if those muscles can't get the signal telling them to contract, they don't. And then the effect then is weakness. So like I said, most nerves are like the ulnar nerve. They're mixed, so they carry both sensory information and motor information. But they can also be mixed in terms of carrying somatic information, so causing voluntary contraction, or sensory information about the body, and then autonomic motor output, which autonomic, remember, that's going to cause involuntary muscle contraction. So things like contraction of the smooth muscle around the stomach, the intestines, that kind of stuff. Or nerves can be strictly sensory, so afferent, or strictly motor, so efferent. And again, we'll talk about some of those when we talk about the cranial nerves next time. In terms of the structure of a nerve, so the basic structure is you've got the axon of a neuron, and so you can see it's a little myelin sheath here, and then that's going to be wrapped in a layer of connective tissue, which is called the endoneurium. So each individual axon is wrapped in a layer of loose connective tissue called the endoneurium. Then groups of axons are bundled together into a fascicle that are wrapped in a layer of connective tissue called the perineurium. And then the entire nerve is wrapped in another layer of connective tissue called the epineurium. So superficial to deep, it is epi, peri, endo, which hopefully sounds really familiar because that's the same basic structure we saw in a muscle. So remember, an individual muscle cell is wrapped in the endomysium, and then a group of muscle cells together is called a fascicle. So again, same concept. And they're wrapped in the perimysium in the case of a muscle. And then the whole muscle is wrapped in the epimysium. So if all the muscle stuff made sense to you, hopefully it did, the structure is basically the same in a nerve. All right, and then last slide. Last but not least, nerve regeneration. So what happens when you injure a nerve? Unfortunately, if that nerve is in the central nervous system, I say nerve, what happens if you injure a neuron? It's probably a better way of thinking about it. If that neuron's in the central nervous system, it is probably not going to regenerate. So I mentioned way back in chapter 12 that once neurons mature, they don't typically divide, and so then we can't usually replace ones that are damaged. And so we talked about that in chapter 12 in the context of things like Alzheimer's disease or alcohol-related dementia and those kinds of things. But let's say you get damage to the spinal cord, which again is part of the central nervous system. So you get some kind of a crush injury to the spinal cord that damages the axons that are traveling up and down the spinal cord. And so once those axons are damaged, they can't relay signals away from the brain or back to the brain. And so that results in paralysis, right? So you can't contract the muscles because they can't get the signal causing them to contract. And then you also get loss of sensation, right? So you can't feel any of those areas. And it's not that the receptors in the skin are malfunctioning, it's that they can't get the signal back up to the brain to be perceived. So if there's damage to the spinal cord, the neurons in the central nervous system typically do not regenerate. And there's a couple of reasons for that. So remember that in the central nervous system, myelination is performed by oligodendrocytes here. And so oligodendrocytes, while they're really good at myelinating those neurons, and that's really important work, one of the downsides to them is that they have growth inhibiting proteins as part of their membrane. And so when that axon of that neuron in the spinal cord is damaged, the oligodendrocytes, the cells that are myelinating that axon, actually keep it from repairing itself. And the astrocytes also play a role in inhibiting that repair. They essentially lay down scar tissue. They're trying to help, but they lay down scar tissue, and that keeps those axons from repairing themselves. So in general, if you get damage in the central nervous system, so damage in the spinal cord, those neurons are not able to regenerate themselves. However, in the peripheral nervous system, because we have a different type of cell that myelinates the neurons in the peripheral nervous system, remember that's a Schwann cell, those cells, those neurons in the peripheral nervous system are better able to regenerate themselves. So the kind of the general way of thinking about it is the farther the damage is from the cord to the neuron. So if you damage a neuron or part of a neuron out by your wrist as opposed to closer to your shoulder, the farther the damage is from the spinal cord, the more likely it is that that axon will be able to regenerate. And actually, whereas the oligodendrocytes inhibit the repair of the axon, the Schwann cells actually facilitate the repair of the axon. So what happens is if you get this crush injury, I'm kind of taking you through this graphic here. If you get a crush injury to the axon here, you can see all the little fragments, and you can see that our Schwann cells have been damaged and our myelin layer has been damaged. That damage to the Schwann cells, they release a chemical that stimulates or that attracts macrophages. So macrophages, pictured here, are a type of white blood cell that'll come in and clean up damaged tissue. So they'll come in and they'll clean up all those fragments, and they'll clean up any of the stray myelination, etc. And then the macrophages in turn release a chemical that stimulates the Schwann cells to proliferate, to make copies of themselves. And so then the Schwann cells are going to proliferate, they're going to make a bunch of copies of themselves and form this regeneration tube that you can see here. And that forms a sort of a scaffolding that allows for these little axon sprouts. And so they sort of protect that regenerating axon. And then eventually, we're able to fully regenerate that axon to lay down all of those little filaments that help us transport proteins up and down the axon. And then we form a new myelin sheath with our Schwann cells, and then our nerve is hopefully eventually good as new. Like I said, neurons in the peripheral nervous system can regenerate, but it's a pretty slow process. So there's a rate that's quoted in the book, 1.5 millimeters per day. But the gist of it is, typically if you get damage to the peripheral nervous system, repair usually takes a few weeks, potentially a few months. But it also depends upon the extent of the damage. So being able to repair this axon is dependent upon having the two ends relatively close to each other. So we only have to lay down a few new Schwann cells to make this neural tube to allow for that sprouting. If these two ends are really far apart, the nerve is kind of pulled away from itself some, then we're not able to bridge that gap. And so then that nerve may never regenerate. So you may permanently lose sensation or permanently lose motor function to wherever that nerve goes. All right, so today was a long one. Sorry, I was thinking it was going to be shorter. But that's the first part of the peripheral nervous system. Next time, we're going to do the cranial nerves, and we'll probably get into some of the spinal nerves some as well. So we'll see you next time.